The overall goal of this procedure is to produce high-quality acute brain slices. This is accomplished by first extracting the brain from the animal. The second step is to dissect the brain region of interest and attach it to the slicing stage while keeping it moist with slicing solution. Next, the brain is submerged in physiological temperature slicing solution. The final step is vibrotome slicing of the brain. Ultimately, the slices can be used in various in vitro electrophysiological experiments.
The main advantage of this technique over existing methods like ice-cold slicing is that it is simpler and produces superior results.
Generally, whoever is new to this method will struggle because it requires all steps to be conducted smoothly and rapidly without unnecessary delays.
We first had the idea for this method when we needed to get high-quality cerebellar nuclear slices from adult animals. The method will be demonstrated by Ankri, a student in our laboratory.
Before starting this procedure, ensure that all tools and solutions are prepared beforehand. Table 1 shows the composition of the standard physical solution, or SPS, in which the brain will be cut. After preparation of the SPS working solution according to the written protocol, equilibrate the solution with 95% oxygen and 5% carbon dioxide for 20 minutes. While the SPS equilibrates, organize the tools required for slicing. This figure shows the arrangements of the tools used for brain slicing and can be used as a visual reference when setting up. The key to identify each item is contained in the written